welcome back to the NPTEL lectures on bioelectricity. So, in the last class I introduced to the curriculum what I am going to follow, and I talked to you about the 5 modules under which this course be dealt with, and I also highlighted the fact that each of the modules will be standalone. So, you really can pick up any module and go through it. So, today what we will do is I will give you a graphical outline of the way the course will progress. So, that will kind of give you an idea where everything fits in, okay? Because whenever we do a course, kind of we lose track, like what is the central theme, how this whole course is being structured, and especially in a field like bioelectricity, which is so diverse from inanimate object to animate object, the insect world, the world of plants and everything. So, where this all these things kind of converge, where really why we devote time. So, today's class will be kind of giving you an overall outline, the flow diagram, how we are going to progress. Of course, your 5 modules will remain there, those are the 5 modules we are going to follow, but all those 5 modules can be put under one graphical representation, and that graphical representation is extremely essential for you people to kind of appreciate, or the integral theme, or I should say the central theme behind this whole exercise of 40 lectures, where kind of you should be able to correlate each and every component of nature, and either you can go on a very fundamental research, fundamental studies about it, or based on the fundamental findings you can think of how this could be used for different kind of applications like biosensors, or prosthetic devices, or bioenergy, plethora of application all over the place. Okay. So, the way I am going to put the graphical representation, let me give you a verbal idea about it. So, first of all, it will be in a tabular fashion, we will depict it. So, in the leftmost column we will talk about the system which you will be studying. It could be an inanimate system, it could be insect system, it could be the systems of animals, it could be a plant system. Then we will talk about the examples under those headings. Then we will talk about the exact physiology or the exact bioelectrical aspect we are going to study in that aspect, and what are the techniques which involved in those kind of a study, and result of those studies where we can take it. Okay. So, overall starting from the basic, identifying the system, characterize the system, understanding the mechanism of the system, and the tools employed into the system, and last but not the least is where we can take it from there. So, let me start with slowly drawing the chart with you. At every quarter I will stop and I will give you my opinion on it, and let us get the whole graphical sketch of the whole course. Okay. So, coming back to the, so this is where we are, bioelectricity, this is lecture 2, and the title of this class will be the graphical representation graphical representation of the diverse bioelectrical events and their implications. Okay. So, to start off with, as I told you, so will be the system identification. That will be our first system identification. This is the first step. After the system identification, we will move on to bioelectrical phenomena of that system. Next, we will talk about the measurement techniques, how we are, measurement techniques, and then we will talk about the advanced applications. And I expect you people to kind of keep this. So, what I expect is that I want this to be act as a guiding principle or should be part of your brain map. So, whatsoever in the next rest of the classes, what I will be teaching rest of the course, you should always try to correlate it with where exactly all those small pieces of information or puzzles are fitting. So, at the end of it, you should have a very holistic picture of nature, how nature is surrounded or pretty much bioelectrical phenomena is should say an integral part of the evolution of nature itself. And whatsoever we study in basic physics, they are all over the place in biology, all over the place. It is just we have to identify the system and kind of quantify the system and establish a link with the existing laws of nature which have been governed in physical world. Okay. So, coming back to the first system identification part. So, this is our system 1. So, basically we will be talking about inanimate object as the first one, inanimate objects of biological origin. So, in this context, I will take you back to some of the most fundamental studies which were done almost, I should say, 40 to 45 years back, while there were some people who were studying the thermoregulation behavior in the hornet nest. So, all of you must have seen a hornet nest at some place or other, this is the hornet nest which kind of in the corner of the buildings or somewhere. And a very intense study was taken up somewhere in the Mediterranean in Israel by Jacob Ishay and other co-workers to figure out how these different nests of nature maintain, regulate their temperature. And in that journey of last 40 years, what all has been discovered, what are the different bioelectrical phenomena. So, in this the example will be, the first example out here will be temperature regulation, in hornet nest. Okay. So, the bioelectrical phenomena is out here is thermoelectrical event. So, talking about thermoelectrical event, so we will be talking about some of the very basic events of nature like Seebeck effect, Peltier effect. So, these are some of the most fundamental properties where thermal energy is being translated or transformed into electrical energy, and vice versa, electrical energy is dissipated in the form of thermal energy. And there are materials which show such behavior, and those kind of materials are being utilized for refrigeration, for active cooling, and likewise there are several other applications of such devices. So, nature indeed has such interesting mechanisms to regulate temperature. So, the bioelectrical phenomena which we will be dealing in the hornet nest is relevant to that. What are the different current and voltages, how they are regulating temperature and all those things. And one of the instrumentation what will be involved in it basically will be talking about the electrometers, which has the ability to. So, talking about the electrometers. So, electrometers are devices which can measure current of very, very low amplitude, very low magnitude like picoampere, nanoampere, with high end fidelity they can measure it. So, we will be talking about electrometers. So, now if you look at it in perspective, so if you look at it, that section 1 falls under all the different phenomena, section 2 falls under the different events, and section 3 is about the instrumentation, because they are all interlinked with each other and the advanced applications of it. So, advanced application of it is one of the applications is inspiration for sustainable buildings So, this is one of the major inspiration using biocompatible thermoregulatory mechanism. So, here I wish to highlight something. So, most of us who live in tropics or temperate countries, during the summer we are totally dependent on an air conditioner. So, which basically essentially does only one thing, it pulls out the air molecules and reduces the collision and between the air molecules within a room using a very strong pump. They sucking out air and thereby reducing the collision and making the room cooler. That is what an AC does. Now, think of a situation and this process of pulling out air from a room needs enormous amount of energy. These are all high power devices. Now, think of a situation if you can replace the amount of energy which it needs could be replaced by something which can maintain the temperature of a room at within a very comfortable biological regime, how that room will be, how our life will be. So, these are some of the inspiration which we derive from nature and they intensely involve a whole lot of biological phenomena. So, this will be our first topic what we will be dealing with under the heading of system identification, bioelectrical phenomena, measurement techniques and the advanced applications, the imagination. Okay. Tomorrow what will happen we do not know, but if we keep on doing this intense research, one day the world may be a very different place. Okay. So, let us move on to the next one exactly under the same heading. Okay. So, again the system identification, I will just put system ID as the first column, then bioelectrical phenomena as the second column, then techniques, and I am just putting the short form, and advanced application AA. Okay. So, the second thing we will be dealing with will be the world of insects, what we learn from them. So, talking about insects, insects have evolved or probably one of the most evolved species on the floor of earth. They have survived millions of years of turbulent weather, turbulent climatic condition, geological changes, and they still survive. And they have adapted several mechanisms of energy conservation, energy harvesting, and several innumerable known and unknown survival strategies in order to ensure that they survive the onslaught of time, the changing time. So, in that process, some of these insects, especially some of these hornets, have developed certain mechanism by which they can trap sunlight and convert it into energy exactly the same way a plant does. So, in other words, within their body, especially within their wings and in the abdomen, they have certain specific molecules which functions as solar energy trapper. They can trap the solar energy and they can convert that, and in other words, essentially what we are talking about, they are living mobile solar cells. So, we will be talking about these kind of solar materials which are found in nature, which has the ability to convert light energy into electrical energy. Okay. So, especially we will be talking about in the hornet, and we will be talking about the solar cells in hornet. So, in other words, here we will be talking about the phenomena will be solar energy to electrical energy. And of course, the different techniques which will be used will be mostly electrical measurement, electrical signal measurement, sorry, and apart from it we will have light signal quantification. And among the advanced application, future energy harvesting tool, energy harvesting I should say strategy. Okay. So, we will pick up one example from the insect kingdom, and we will talk about how the insects harvest energy from nature. And this is something which could be a big inspiration for the future, where as all of you have seen that silicon industry is almost hitting the roof. Crystalline silicon's efficiency is around 17% and 17 to 20%, maybe maximum, that too in the lab conditions. So, there is intense search for molecules which are much easily available, synthesizable, more green and more biocompatible, which does not need the extensive cost of processing silicon. So, in that line, one approach, inspiration from nature, is what I just now explained, is lies in the cuticles of the hornets, and if we could synthetically develop those compounds, they may have immense potential to look forward. Okay. So, let us identify the third system from here. Okay. Again, there will be 4 columns this time, again the same thing, system identification, the column 1 SI, system identification, the biological or bioelectrical phenomena, the instrumentation, and advanced applications. Okay. So, here we will be talking about the world of plants, another very, very diverse kingdom, and the only known biological system which with its fullest ability could harvest sunlight. gets the light, convert it into electrical energy in terms of flow of electron, and helps to synthesize food. So, in the food chain, they are the autotrophs. Auto means self, troph means they have the ability to synthesize food, and we all depend on these autotrophs for our livelihood. So, they are kind of the pillars of the ecosystem. Based on those pillars, the whole food chain is dependent upon. So, that is one of their most fundamental contribution. Apart from it, they have wide range of sensors in their body. They have touch sensors in the form of Mimosa pudica. They have touch sensor as well as mechanical, you can say touch sensor coordinated with a mechanical door in terms of Venus flytrap, where they could trap an insect. But that whole mechanism by which biology ensures a fantastic micro electromechanical system, which all of you comes with the name of MEMS. So, biology already had these kind of MEMS inbuilt in their system. So, that is an inspiration for those who are working in the domain of MEMS or miniaturization or mesoscopic devices. It is a big inspiration. So, that is the third thing we will be dealing with. Apart from it, the whole world is so beautiful because of the beautiful flowers all over the place, and these beautiful flowers does something very amazing. Actually, they are the inspiration for dye-sensitized solar cells. Those of you have heard like Gretzel cells after the name of Gretzel, who has done pioneering work in the field of dye-sensitized solar cells. So, what are those different different components of nature which contribute to develop different kind of dye-sensitized solar cells? Because that is, as I was telling you in the previous slide, that silicon technology is hitting the roof. So, we all are looking forward for next generation. So, dye-sensitized solar cell is another one in that line. So, different ruthenium dyes, different floral dyes, and all those things which are derived from nature, which is basically an inspiration to develop the next generation of high-end dyes. So, talking about the plant kingdom now, that is what I have just narrated you. So, plant kingdom. Okay. So, within the plant kingdom, we will have A. We will be talking about photosynthesis. Then we will be talking about the Mimosa pudica or touch me not for touch sensor. This is for light to electrical to food. Okay. Then you have Venus flytrap, which is basically a BioMEMS trap. Okay. And then we will be talking about dye sensitized. So, the flowers, flower dye for dye sensitized solar cells. Okay. So, the instrument here, there are multiple instruments which are being used, which are fairly common. Of course, you need electrometers, amplifiers, you need a whole lot of biochemical techniques to study photosynthesis, and you need the high-end MEMS, micro electromechanical systems, and likewise several other instrumentation which is needed. And among the advanced application in terms of photosynthesis, we are talking about artificial leaf. I am coming to this. What does that mean? So, say for example, So, we talked about photosynthesis. So, essentially what is happening on the leaf surface, light is falling and this light energy ejects an electron, a photon is being absorbed, ejects an electron, and this electron through the cascade of through a pathway along the chloroplast leads to the formation of glucose molecule, which is being consumed by our body. And of course, it goes through photosystem 1, photosystem 2, and the output of this is you are splitting a water molecule to oxygen as a byproduct and you are evolving hydrogen. So, there are two inspiration here. The first inspiration is that making synthetic chlorophyll molecules, which could be used for trapping solar energy and injecting an electron. That is one approach. The second approach is that, so coming back, okay, the second approach is that whatsoever we get inspiration in terms of the manganese cluster for splitting water. So, let me talk about at this line, let me make one correction here, and if this is one aspect of it, there is another aspect of it, which is water splitting, and this is all taking place within the domain of photosynthesis. Okay. And artificial leaf, or we can add something else here also, synthetic chlorophyll for solar energy, artificial leaf for water splitting. So, understanding photosynthesis and emulating photosynthesis could have profound impact in our understanding of nature, and this is one of the most fundamental reactions of energy conservation, which is being followed by nature. So, we will go in depth into photosynthesis and we will talk about it, and from there we will move on to the water splitting cluster, which is part of the photosynthetic machinery. Okay. So, then we talked about mimosa pudica or the touch sensors. Okay. So, these touch sensors, they have been studied for almost last 100 years, and some of the pioneering studies were done by Sir J.C. Bose, Jagadish Chandra Bose, and he made some very pioneering contribution, and we will talk about his contribution, where he talked about whether plants have nervous system or not. As a matter of fact, in some of his philosophical transactions, he has kind of hinted upon that these are rudimentary nervous systems of plants which could sense touch, and we will talk in depth and what are the different devices which are being used and what is the current status of the field, and which could be an inspiration for developing touch sensor for the future. Okay. So, we will talk about J.C. Bose's contribution and talking about touch sensor, and while talking about these touch sensors, I wish to highlight one more point, that he is among those very first people, Bose was among those first people who could show a functional semiconductor device in the form of galena. We will talk about it a little bit more. Okay. So, now coming back to the Venus flytrap here. Okay. Venus flytrap is a kind of a device which has an inbuilt touch sensor as well as a mechanical event. So, whenever a insect comes and sits there, it stimulates some part of the flower, and there is a hood which closes in like this. So, in other words, this is a touch sensor coupled with a mechanical door, touch to mechanical door, and this whole connection could be a big inspiration for future for developing MEMS based devices, and which is purely an electrical and mechanical phenomenon. Okay. So, we are talking about here a touch, okay, touch coupled with mechanical action. This is what Venus flytrap will be studying, and then we will be talking about some of the contribution made by Professor Gretzel, who is among the pioneer in developing dye sensitized solar cells or Gretzel cells, which is commonly known. So, we will be talking about some of those dye sensitized solar cells, and basically talking about talking about some of the contribution of Professor Gretzel and his Gretzel cells, or one second, sorry, and we will be talking about the energy harvesting. And in the previous slide, while I was talking in previous slide, while we were talking about inanimate objects of biological origin, I'll have one more column which I haven't mentioned. So, if this one is the first one, so we talk about this is the second one, which is the world of insects, and we talk about the third one, insect plant kingdom. So, there are two more which are left actually. I haven't talked to you about the kingdom of animal, which I'll be talking in the next class, and there is one more section which I'll be introducing, which is not very clearly highlighted in the course part, is about some of the very primitive inorganic molecules, which has some amazing electrical characteristics, which has wide range of implication in biological processes, and we will talk about those. So, I will close in here today. We will continue here with the same graphical scheme of things in the next class, and then we will move on to the different steps of the course. Thanks a lot.